Welcome, Biochem students, to Snow Day Chapter 16 material. This is the first slide with the objectives, and just since we haven't had class, I wanted to kind of refresh where were we. We're in lipid metabolism, and we were looking at beta oxidation. That's the fatty acid breakdown. Thursday after the quiz, we'll do fatty acid synthesis. But for now, we're going to focus on just a quick recap of breakdown, and then we're going to look at odd chain fatty acid breakdown and unsaturated fatty acid breakdown. At least that's the goal for this video. Okay? Oh, let's discard. Alright, so this is slide 15. You've already seen this slide, but if you recall, we had a fatty group attached to coenzyme A, and then there were a variety of colors. This happens to be a 16 carbon, and then for each round of beta oxidation, you chop off 2 carbons, leaving you with 2 carbon units on the right each time. Okay, so this was sort of a summary slide going through the entire process, and you end up with a whole bunch of acetyl-CoA molecules as your products, which is why when we did the net reactions, we looked at the net for one round with acetyl-CoA that goes into the citric acid cycle, and that gave us ATP. And then this net reaction we already saw. This writing one will be on the exam, but writing the net reaction for one round is going to be on your quiz this Thursday. So that's why I wanted to show that to you. So you remember we have a fatty acid attached to CoA. There's going to be FAD, NAD, CoAsh, water involved, and then pair FADH2, NADH plus H plus, and then we have the appropriate, just do your math for your acetyl-CoA coefficient. I'll show you the math in a minute, but we start with 16 carbons. If acetyl-CoA has 2 carbons, there better be 8 of them. That's, like I said, for the exam for next week. And we already looked at this slide, so I'm going to skip ahead. Okay, so I want to look at total ATP from a certain chain length given, because we did calculations in Biochem 1 of ATP from 1 glucose. You have to do that for me on the next exam, ATP from a given fatty acid chain length. Okay, so I'm doing the same palmitic acid, 16 carbon fatty acid. So these are the net products from the previous, or 2 slides earlier, from the full breakdown of 1 16 carbon palmitic acid. Okay, so you can do the math as I show here in the bullets, and you get 108 ATP. Now you might not remember this, but we had this whole process called activation, fatty acid activation, that we dwelt on last class, and that used up, it was equivalent to 2 ATP. So I can't say I get 108 ATP. I need to subtract 2 from the formation of the original activated fatty acid, because remember, it used to be attached to glycerol. So the final actual number is 108 minus 2, which is 106 ATP. So this is your example. You can pick any length you want, but you're going to have to do it on the exam next week. Okay? One thing I do also want to compare is if that was 106 ATP from palmitic acid, if you actually look at the math for the energy produced, you get way more energy in kilojoules per mole from the degradation of a fat than you do from the degradation of glucose. And so that's just, it's giving you more energy. And if we were in class, I'd ask you why you think this, and we'd have this whole long critical thinking discussion about it. But since I'm talking to my computer, here we go. So essentially, the carbons in the fat are starting at a lower oxidation state in the fat than they are in glucose. And so, therefore, you get more energy as a result. Okay, this was the math I was talking about, once again, for even-numbered carbons. If you're trying to figure out how many coefficients are going to be on your acetyl-CoAs and your NADHs and stuff in your beta oxidation, you're going to have to do the number of carbons. If that's X, then acetyl-CoA is X divided by 2. So 16 carbon fat divided by 2, be 8 acetyl-CoAs are formed. Do the same thing, except you subtract 1 for the rest of them, the NADH, QH2, or FADH, whichever you want to call it. It's the same process. 16 divided by 2 minus 1 is 7. So that is all for even numbered fats. Okay, even numbered fats. We're going to spend several slides on odd numbered fats, odd chain fatty acids. Okay, so this would be like 17 or something. So remember, we'd go all the way down the same way. You'd actually start the same way. Beta oxidation is the same until the last cycle. Okay, chop off your 2 carbons at the very, this goes away and he's happy as our acetyl-CoA. But at the very end, we have these 3 carbons and we can't chop off only 2. So that leaves us with this molecule, propionyl-CoA. What a great quick multiple choice question for Thursday's quiz. This Thursday's quiz. Okay, from there, I'm going to number the steps 1, 2, etc. for odd chain fatty acid breakdown. So we start with propionyl-CoA once we've done all the other acetyl-CoAs as was performed for even chain beta oxidation. Okay, so the first step is propionyl-CoA plus, sometimes that's written as CO2, sometimes it's written as HCO3 negative. Okay. And we form methylmalonyl-CoA, okay? You may see some books call this SD-methylmalonyl-CoA. It's the same thing. It's just a chirocarbon. So one book will say S, one book will say D. It doesn't matter, okay? Biotin, which is a common vitamin you can take, biotin is a big cofactor for that process, okay? This is showing the image from your book that is the carboxylation for propionyl-CoA, okay? And so here is what was CO2, but in the cell it can be HCO3 negative, the bicarbonate, it's the CO2 there. One of the things that I'm going to have you do on the quiz is I'm going to give you a reaction kind of like this one, and you need to tell me what kind of chemistry is happening. You need to be able to look at these structures and say, I'm not really sure what's going on, all of the chemistry, especially because biotin's a pretty ugly looking structure, but I can observe that I have gained a carbon, okay? At this point, you guys should be able to look and not be scared of structures like the propionyl-CoA and the methylmalonyl-CoA, okay? So, the next 3 steps, I'll go ahead and give you, I'll go ahead and give you all of the slides. The next 3 steps, you're sort of looking at conversion to succinyl-CoA, eventually to succinyl. So here's the methylmalonyl that we ended with on the bottom of the previous slide. Isomerize it and do another reaction with B12 as the cofactor, and we end up with succinyl-CoA, okay? So that's, like I said, 3 steps produce succinyl-CoA. That's steps 1, 2, 3. From there, our succinyl-CoA, okay, is converted to succinate, malate, pyruvate, and then acetyl-CoA. All of these are common either citric acid cycle or glycolysis intermediates, okay, steps 4, 5, 6, 7, and 8. Okay, so those are really simple. They can go in and out of the citric acid cycle. Your body can use these intermediates very readily, okay? So that's pretty simple and easy and straightforward. Okay, I'm going to show you the reaction 2 real fast. So here's your S to your R. Or the enzyme is a racemase. It's an isomerization. Okay, nice and simple, easy reaction. I don't think I have anything more to add to that. If you wanted to, you can make a note. This would be D and L if you were using DL configuration. Reaction 3, methylmalonyl-CoA to succinyl-CoA. This is what I already showed you a couple slides ago. Okay. And you should recognize succinyl-CoA. That was in the citric acid cycle last semester. Okay. Here's the cofactor known as B12. It's also called cobalamin. I'm not sure how to say that correctly. Okay. So it has a cobalt. You should be able to look at this and note, wow, it's got like this huge heme group and some other structures that look kind of like nucleotides. So really complex, but not so scary anymore, okay? And that's just a fun fact about that particular enzyme, okay? Because the actual reaction generates a carbon radical, and so this cofactor helps stabilize the mechanism for that reaction. So it's kind of neat, Okay, so I'm going to zoom in and show you reactions 4 through 8. Here's 4 through 6. You don't even need to know them all. Eventually you get to malate. Okay, we're using a lot consumed produced materials. Okay, the next are 2 decarboxylation steps. This is where if I tell you that, look at them and be like, oh, I can totally see that. I can see the decarboxylation going on. Okay, I have CO2 produced here. I didn't show it coming out of my reaction like that. Okay, NADPH is also produced. I didn't show that arrow on my diagram. Reaction 8, another decarboxylation, leaving us with our beautiful acetyl-CoA. Whoops, acetyl-CoA. You're going to find he's like the MVP of fatty acid lipid stuff. Okay? So now we have our happy acetyl-CoA. He can go on to the citric acid cycle. So it's a lot of complex steps from the propionyl-CoA. All right, unsaturated fatty acids. This is one other thing I want to mention. Normally, fatty acids exist in the cis conformation that's shown here at the top, okay? So if they're in the cis, then an additional, or excuse me, normally they're in the trans, but if they are in the cis, then all we have to do is isomerize it from the cis bond here to trans. That's it. It's one step. That's an isomerization. And that's all you need to know from this slide. If we happen to have cis, we got to switch it with an isomerase to trans. You don't need to know the exact name, just that it's an isomerization reaction. Okay? So for an unsaturated fatty acid, if it's an issue, isomerize it. Okay. I want you to write down this information, and we will probably start on the next slide after the quiz on Thursday, but this way you can at least have this information, because biosynthesis we're going to dwell on for a little bit. It's got some prep work just like activation for breakdown did, but it's a huge enzyme, fatty acid synthase, that has a bunch of different subunits. If you see, they all have these little codes on the diagram, okay? And fatty acid synthase is just like one huge enzyme that does the full biosynthesis. It's a pretty neat enzyme. So we'll talk about that. For the quiz, you definitely need to know the net reaction for beta oxidation for one round, some facts about odd chain fatty acid breakdown that we just talked about in the video. Okay. Other facts about the activation process, like the carnitine pathway, the stuff we talked about last week. Okay. And like I said, there's one more question where I give you some structures and you kind of got to look at them and tell me what's going on in the diagram. Think like a biochemist. Be able to look at them and not be scared of them. So I hope everybody stayed warm on your snow day, and I'll see you in class on Thursday.